Hi everyone, this is Mr. Sinti again, and this is a follow-up video on enzymes, which is a special kind of protein. And enzymes are going to be very important to us this year in biology. And one of the things about enzymes is that they're proteins, and so let's consider this idea of protein. So this thing here in green is a cartoon drawing of a protein, and although I think it's pretty good. Do you see these things that sort of look like green grapes? All of those are amino acids, and I like this picture because it isn't just a straight chain of amino acids. Like if you're thinking that a protein is made up of chains of amino acids, each one of those is an amino acid. Sometimes a string like this doesn't give you the true impression of a protein, because what happens is the protein can fold back and form all of these interesting three-dimensional shapes. And so what's interesting is you get all these amino acids forming, and it results in a three-dimensional molecule that has, and let me emphasize this particular area, it's sort of like a baseball glove. Like this area right in here is like a pocket where the ball fits in. And this area where molecules can fit inside of a protein is referred to as the active site. That's rather important. And it's like, oh yeah, what about it? Well, when a chemical reaction is taking place, it can take place inside of an enzyme. And what the enzyme does is that it provides a small, sort of a small atmosphere, an alcove, a space, a crevice where substrates can collide. And it's all about collision in a chemical reaction. The molecules, sort of like cars, they need to have speed in order to really make an impact. If they're crashing, if they're going slow, they're just going to bump into each other. And so temperature can be harmful or deleterious to cells in general. And so enzymes provide a space where molecules can meet up at lower temperatures, and therefore they could speed up a chemical reaction and act as a catalyst. A catalyst is something that speeds something else up. Let's investigate this further. So if you look at this next slide, well, in the previous slide, do you see this spot right here, this sort of red looking object? Let's say that that is a disaccharide called sucrose. It's a simple two monomer molecule, sucrose. It's sometimes called table sugar. And I can ask you these questions about it, and you're like, okay, how is a disaccharide like sucrose digested? And you're thinking, wow, very simply. All you need to do is add water, and water comes in and breaks it into two monosaccharides. And so the answer to that is hydrolysis. And you're like, okay, so if the reaction breaks up into glucose and fructose, they must be the products, and they are. Glucose and fructose are the products of sucrose being broken down. But this is the key to the whole conversation that we're having right now. The reaction's very, very slow. So in other words, if I were to say, okay, here's sucrose and here's water, what am I expecting? I'm expecting glucose and fructose. But I may be expecting it, but I'd have to wait a long time. And you're like, come on, I want this chemical reaction to occur more quickly. Well, one way to make it occur more quickly is if you put the sugar in a glass of water and then heat the water or boil the water. In boiling water, the sucrose and the water will be colliding, and you'll be able to get a hydrolysis happening really, really quickly. But there's a problem with boiling water in our body. Our body doesn't like boiling water. And so here's the thing. In a protein, you can get sucrose and water to meet up in this small little area called the active site. And so this active site is rather important. This active site right here, active site, is this area of the protein. The whole protein's not shown. If I do this, maybe it is. This allows these two molecules to collide. Now what are we going to call these? We're going to call the molecules that collide substrates. So the substrates fit into the active site, they collide, And what's interesting about an enzyme, there's many interesting qualities, but they're very, very specific. And it's kind of like a lock and key. This pocket, this is where the analogy of a baseball glove kind of comes undone, because as you know, a baseball glove is supposed to catch baseballs, but it can also catch oranges or lemons or a softball, or catch really anything kind of round. But enzymes are extremely specific, which means that only that substrate is going to be able to fit, and so the enzyme is only going to speed up certain chemical reactions, and so therefore enzymes are able to overall regulate the rate of all metabolic reactions, kind of like traffic signals in a city. They're not all green or all red. Some of them are going green, some of them are red. So by being specific, they can regulate particular chemical reactions as necessary. That's a very important point, and it's something that will be discussed throughout the year, so don't worry too much about it, not understanding that completely. This is just the basic of how an enzyme is working. See this big purple thing? Here's our enzyme. It's made up of, it's a protein, which means that it's a big chain of amino acids, but notice how it has a three-dimensional shape. Do you notice how these little pockets right there? That's the active site. And what's going to fit in? What's going to fit in is the substrate, and in this same example, it's table sugar. So the sugar goes into the active site. Who else goes into the active site? Well, water needs to go in there as well, because water is what's splitting it up. So when water splits it up, the products pop out, fructose and glucose. Now, I like this picture a lot because can you see right here how the fact that the enzyme hasn't changed its shape? And so it doesn't directly participate in the chemical reaction. It speeds up the chemical reaction. So it's sort of a helper. And so it could be used over and over and over again. In other words, the substrate goes in, the water comes in, it splits the molecule up, and the products come out. And so enzymes are specific, and they can be used over and over again. That's an important property of an enzyme. Couple things more to say about them is naming them. You want to have a system of naming, just like people. When there's a lot of people, you want to be able to call them by name if you're interested in saying something about them. And so enzymes have names, and what we do is we try to use the name of the substrate, get rid of the suffix, and add the ending ase to the name of the enzyme. So for example, if we're trying to digest sucrose, the enzyme that speeds up the digestion of the disaccharide sucrose is you remove the ose, right there, let me cross it out, and you put ase. And so the name of that enzyme is sucrase. So sucrase helps to digest sucrose. This is a very famous enzyme right here. The one that digests lactose is called lactase. So if you're lactose intolerant, you're lacking, pun intended, you're lacking lactase. So you don't have that enzyme, and so therefore you're having a difficult time digesting it. And then maltose is maltase. Let's see if we go to the next one. Now the last thing that I want to say is some characteristics about enzymes. They're proteins, they're very specific. I mentioned that they're reusable because they don't actually get consumed in the chemical reaction. They just provide a space. And oftentimes, detail, they don't work alone. They have little non-protein helpers, like for example vitamins called coenzymes that help them work efficiently. Now overall, you can say that enzymes speed up chemical reactions, but what they're really doing is lowering the need for activation energy. And activation energy, you can think of it as like a match. So if you want to get paper to ignite with oxygen, you light paper on fire, paper is sugar, oxygen's in the air, it's gonna burn. Paper will turn brown, but it's a slow process. An apple will turn brown, sugar and oxygen, but it's slow. The reason it's slow is because the molecules are moving slowly, and so what enzymes do is that they provide a space that molecules can collide without a lot of activation energy, without a lot of temperature. And so speaking of temperature, a factor that would affect enzyme activity is temperature. If molecules are moving really slowly, do you see this at cold? Things are going slow, that means that the reaction's going to go slow. Room temperature, a little faster, a little faster, a little faster, and you're like, wow, this is really fast. What's that? Body temperature. So 37 degrees is optimal for most enzymes to work fast. You're like, well, gee, 37 is going to make it go faster. See, the trend is in an upward direction. You're like, well, I'm going to just crank it up to 50. Maybe it'll be really fast. And you're like, if you crank it up to 50, what's going to happen is it's so hot that it's going to move the protein around so much that it's going to lose its shape in a process called denaturing. And if it loses its shape, it's going to lose its functionality. And so this is kind of important. In really hot environments, enzymes don't work really well at all. And so this is of concern when someone's running a high fever. When you're running a high fever, you have to be really careful, because if it's too high, your enzymes will lose shape. They lose their shape. None of your chemical reactions are going to be working. It's not so good. Likewise, pH will also affect the shape of the enzyme. Throw an acid onto it or a base, it's going to change the shape. And so, finally, let's look at this last slide. If you change the shape of an enzyme, like in other words, high heat, it's not going to be able to work properly, because it's its unique shape that gives it the key to its functionality. Okay? And so the last thing I want to say is, in the lab that we're going to be performing, one of the first things that we're going to look at is the sugar lactose. So if you're following this, you'll know that the enzyme is going to be called lactase, and it's going to break it down. It's a hydrolysis. And here's my question that we're going to reveal in the lab. How do you know if lactose is being broken down or not, and broken down more quickly? I mean, how is it? I mean, are we going to be able to see that? How is it that we're going to be able to study whether or not a chemical reaction is occurring faster than it would be at room temperature if you add an enzyme? So this is kind of a cliffhanger. So stay tuned for the lab. I hope those of you who are watching this are at the Giants parade. It's great. Congratulations to the Giants. Awesome. So let's just see if we can shut it down. We'll go over here, and we'll go like that. See you later.